Good morning, good afternoon, and good evening, everyone. And welcome to the Scientist.com and Discovery Life Sciences webinar titled Considerations in Tissue Acquisition and Characterization for the Development of Long-Term Tumoroid Models. I'm Liam Sanyo from the events team here at Scientist.com, and I'll be your host today. We're very fortunate to be joined by Philip McCauley, who's a product manager in biospecimens, and Dr. Sean Fall, VP Lab Operations in Cell Services and R&D and Biospecimens. In their presentation, Phil and Sean will explore cutting-edge 3D tumor models and their potential to accelerate cancer research and development. And without any further ado, I'd like to introduce the first speaker, Phil McCauley. Phil, thanks so much for joining us today, and the floor is yours whenever you're ready.
Awesome. Thank you, Liam. And thank you, everybody, for joining today. Happy to have you here with us. I'd like to begin with the why we're here and give some context to the reason we're discussing the development of these advanced tumoroid models today. The challenge of getting a therapy from preclinical stages through clinical phases is well known. Only about 10% of preclinical therapeutics that have moved into phase 1 trials end up getting approved, with most of those early trial failures due to a lack of efficacy and/or toxicity, exposing a significant disconnect between preclinical promise and clinical reality. This stark gap underscores the need for advanced in vitro models to better predict therapeutic success and translate findings more efficiently from bench to bedside. And as I move into our next slides, I want to start with an interactive poll here. The first question that's going to come up is, what tumor models do you currently work with? I can give you a second here to submit a response for that as I move into our next slides. So let's start with reviewing the history of tumor models and how they have evolved into the powerful research tools used today. So it's the early 1950s. Researchers have just immortalized the first cancer cell line with Henrietta Lacks's HeLa cells as the first patient-derived tumor cell culture, or PDC. This was a game changer. Scientists now had an endless supply of cancer cells to study, and it's led to many discoveries. But as exciting as that was, limitations such as genomic instability and lack of tumor cell heterogeneity made them less predictive of actual tumor responses than future models. By the 1960s and 70s, the focus shifted to mouse tumor models, giving us a glimpse of cancer in an actual living system. But still, mice aren't people. So while these models were a step forward, they didn't always predict what would happen in human patients. Then in the 1980s, we saw the birth of the NCI 60 human tumor cell line screen, which is a panel of 60 diverse human cancer cell lines used by the National Cancer Institute to serve as a standardized platform for evaluating anti-cancer activity of potential therapeutic agents and allow testing across multiple cancer types. So it was really empowering after the development of those immortalized cell lines into something stronger. But still, while it was a leap for the field, they were still limited by those 2D cell culture methods that they were using. So then we get into the 1990s. And that brought genetically engineered mouse models, or GEMs, and xenograft models, where human tumor cell lines were implanted and grown in mice. Now, this was an advancement of the mouse models that were being used and got us closer to a true human-like environment, but still, many tumors behave differently in mice than in people. And they were still using those immortalized cancer cell lines to create the xenografts. So we move forward again to the 2000s, and that ushered in patient-derived xenografts, or PDX models, as well as spheroid cultures. And these were actually generated from primary patient tissue instead of those cell lines. But even with those advanced mouse models and these basic 3D cell culture models like spheroids, researchers still faced predictive limitations. So we move forward again, we get to the 2010s. And we see the rise of 3D culture systems, starting with organoids. Now, organoids are self-organizing multicellular structures that better replicate in vivo physiology. And organoids started with normal tissue, starting with colon tissue at first and moving on to other normal tissues before moving to actual tumor tissue when the first cancer organoids or tumoroids were developed. But there was a lack of a reliable, reproducible source. A lot of this work was being done independently in labs. These were one-off kind of proof of concept research tools, and there wasn't a large source to be able to empower everyone to use them. As well as with the time period, genomic and biomarker profiling at the extensive elements that they are now were just not standard at the time. So finally, we get here to the 2020s, and the field sees the emergence of advanced 3D tumoroid models, as well as biobanks of these model systems. These tumoroid models are generated from genetically and molecularly profiled primary patient cells and more closely mimic in vivo tumor physiology than previous models, giving us unprecedented insights into patient tumor biology. We've also seen evolution in vitro preclinical testing in the field in general, when in 2022, the FDA Modernization Act 2.0 was signed, which removed the mandatory animal testing requirement for new drug development, allowing alternative testing methods, including in vitro assays performed with tumoroids, to be considered for regulatory approval. So it's been a remarkable journey from these basic 2D cultures to these cutting-edge 3D models that more closely mirror human biology. Every step along the way has brought us closer to more personalized, predictive, and effective cancer treatments. And these models continue to evolve, and so does our hope for better outcomes for patients everywhere, as researchers bridge that gap between preclinical studies and clinical trials. So we've looked at the history of the tumor models. Now I want to take a closer look at the advanced 3D tumor model themselves. So tumoroids. Tumoroids are essentially miniature versions of primary patient tumors grown as 3D in vitro cell cultures. They are self-aggregating and multicellular, meaning that the tumor cells that make up the tumoroids are not just one clonal version. They are made up of cells with varying level of mutational burdens and biomarker expression levels, just like we see in the in vivo tumor microenvironment. Tumoroids also overcome logistical challenges. They are cryopreserved, making them convenient to fit into testing timelines. And you can bank them at particular passage points, just like a cell line, allowing for reproducible and comparable testing on the same patient over time. They're also scalable and can be cultured in flasks to generate enough tumoroids for any assay while still maintaining their unique cellular features. All in all, because they more closely mimic the architecture and behavior of the original tumor than previous models, come from reducible biobanks, and are ready for most any downstream in vitro assay, they truly represent an advanced tumor model system. And as we've established, a major cause of failure in clinical trials stems from inadequately predictive preclinical model systems, which do not accurately replicate human tumor biology. So let's compare tumoroids to some of those models we talked about and see just why they're becoming such a valuable tool in cancer research. Traditional models, like these immortalized cell lines that we talked about, fresh primary tissue, animal models, 2D human cell cultures, and simple spheroids, each have significant limitations. For example, cell lines can drift genetically over time, but fresh tissue can be hard to source and maintain. And animal models just don't often translate well to human biology. Meanwhile, 2D cultures and basic spheroids lack the full three-dimensional complexity of real tumors. Because of these challenges, there is a need for more advanced clinically relevant models that can accurately predict how treatments will work in patients. And so unlike those traditional models, which often fail to fully replicate the human tumor complexity, tumoroids do maintain the original tumor's genetic diversity and more accurately mimic its behavior by maintaining that donor-specific characteristics and intratumor heterogeneity. They preserve the tumor physiology in 3 dimensions, so providing a much more realistic and predictive platform for drug testing and cancer research. In short, tumoroids can better bridge the gap between traditional models and real-world clinical outcomes, offering a more reliable way to assess treatment responses and predict patient outcomes. So we've looked at the cancer model history. We've taken a closer look at tumoroids. Let's zoom out a little bit and see how this fits into the bigger picture of tissue collections leading to a biobank and how that enables our ability to generate tumor lines. So we at Discovery have a global collection network that plays a critical role in enabling our ability to generate these tumoroid lines. We source and have created a biorepository representing a wide variety of patient tumor samples. This extensive biobank of annotated, characterized samples, along with our access to fresh tissue, provides us with a diverse starting material source for tumoroid generation screening. So here's how we secure these high-quality, assay-ready biospecimens. This graphic represents the assortment of tissue and biofluid specimens we regularly collect and bank. I'm going to focus on our tumor tissue collections over here on the top. We begin these collections with surgical resections of biopsies from primary patients. And these are often a one-time opportunity to collect these precious, unique tissue samples. Our strong partnerships with hospitals and research institutions around the globe give us access to a diverse range of these well-annotated samples. And of course, we follow strict ethical and regulatory guidelines, including informed consent and IRB approvals, ensuring that every sample is responsibly obtained, de-identified, and ready for downstream applications. So I've mentioned our global clinical network, and to give you some more information on that, our global clinical network spans nearly 200 sites, giving us access to this diverse patient populations of tumor types. This worldwide infrastructure is the backbone of our ability to rapidly acquire and process high-quality tumor samples. By processing these samples in our centralized labs in the US and in Europe, we minimize transport time and preserve tissue quality, both critical aspects for generating reliably clinically relevant models. This also allows us to provide solutions for overcoming the challenges of actually working with fresh tissue. As I mentioned before, fresh tissue has many logistical challenges, and I want to take some time to go over those. So here are some of these challenges of working with fresh tissue. The first thing is timing is critical. Because they're fresh, these samples must be used right away due to their limited stability. They can't be stored long-term to be used when it is convenient. In many cases, because these fresh collections happen right during the resection and before additional health records can be reviewed, details like tumor subtype, stage, even cell yields, populations, and viabilities aren't well known until after additional testing and pathology reviews, which normally isn't done until the timeline required to actually work on the fresh tissue. There's also the possibility of an incorrect diagnosis, a primary site that ends up being a metastatic site for a different indication. And you may not have immediate access to mutational profiles or follow-up patient data. All of these unknowns make fresh tissue logistically difficult, underscoring the need for strong and efficient workflows to ensure we get the most from the samples that we do collect. And one of the ways we provide solutions to overcome fresh tissue logistics and limitations is through our processing of tumor tissue into dissociated tumor cells, or DTCs, which are valuable starting materials for advanced tumoroid models. We dissociate and cryopreserve the entire tumor microenvironment from each tumor tissue collection before adding them to our biobank repository for stable long-term storage. And this process starts when we collect resected tissue through our global clinical network and immediately ship it to our central labs. Once there, they are carefully and proficiently processed through a proprietary enzymatic and mechanical dissociation procedure. We cryopreserve the isolated cell populations of the tumor microenvironment and perform post-thaw QC viability and yield counts. From there, we further characterize the tumor cell population through flow cytometry and unique genomic and biomarker profiling through a variety of methods. This optimized tumor tissue processing approach, coupled with comprehensive characterization from biobanking infrastructure, ensures a reliable supply of high-quality DTCs for downstream assays and applications, including tumoroid generation. So we've looked at fresh tissue, we've talked about DTCs. Let's do a quick comparison here. So DTCs offer a clear advantage over fresh tissue for all of these reasons. They're cryopreserved and available on demand, which lets you bypass the use it or lose it timeline when dealing with fresh tissue. You can now work on multiple patients in parallel at the same time in an assay, as opposed to having to run them one at a time not on your schedule, but at the schedule of the actual fresh tissue acquisition and delivery. Here at Discovery, we actually confirm the pathology from the patient tumor, and we run flow cytometry to assess the cell population composition within the cryopreserved DTC vials, so you know exactly what you'll be working with. Further in-depth analysis, like NGS sequencing, PCR, flow cytometry, etc., strengthens our DTC's biobank, allowing researchers to pinpoint patient tumor samples exhibiting the exact mutational and molecular expression profiles that they require for their research. Overall, DTCs are a flexible and reliable choice for downstream research and enable further advanced tumoroid models to be generated. So to summarize, Discovery, we collect these patient tumor samples, we reliably bank them, characterize them, and then we are able to screen them for tumoroid model formation and to be able to supply a large biobank of these advanced tumor models. I now want to pass things over to Dr. Sean Fall to discuss more in depth about biomarker and genomic characterization as it relates to tumoroids. And as I do that, I'm going to provide another interactive opportunity here with a poll question, this one being, what areas of research would you use tumoroids for? Thank you.
Fantastic. Thank you, Phil, for a great introduction to tumor models and how at Discovery we've really tried to position the availability of starting material to be robust and instantaneous. So for that poll question, we were really focused on the uses of tumoroids and primarily in a immuno-oncology space. We're going to focus on two of these for the second part of the webinar. The first is going to be checkpoint inhibitor therapies. This is really one of a number of numerous immunotherapies that have really revolutionized cancer treatment over the last decade and a half. They were really driven by the first approval of an immunotherapy, which is the anti-CTLA-4 antibody in 2001. And then in 2018, the Nobel Prize for Medicine was awarded for checkpoint inhibitors, particularly CTLA-4 and PD-1, which we're going to discuss more throughout the rest of the talk. The idea behind checkpoint inhibitor therapies really relies on one of the fundamental principles of cancer, which is the acquisition of mutations. So in this case, there are numerous factors. They can be environmental, they can be genetic, which allow for the acquisition of numerous mutations throughout the genome. And this leads to the activation of oncogenes, the loss of tumor suppressors, and eventually leads to transformation in cancer. One of the very nice readouts of this idea of tumor-specific mutations comes from mismatch repair proteins. Particularly, there are 4 that are examined: MLH1, MSH2, MSH6, and PMS2. And a deficiency in any of these can predispose to a high degree of instability within the genome. The very interesting thing about these mutations is that they produce what's known as neoantigens. And so these antigens are unique to the tumor. The immune system is not tolerized against them during their normal development, and therefore it can be seen essentially as non-self to the immune system. And indeed, there have been many, many studies that have shown that there are T cells present within the tumor that show some reactivity to the tumor itself. However, those T cells are largely hyporesponsive. And one of the mechanisms of that is the expression of inhibitory receptors on their cell surface. One of the most best studied of these is PD-1. And so PD-1, when it's engaged, actually turns a T cell off, such that even if it could recognize a target cell to kill, it becomes unresponsive. Within the tumor, there is expression of the ligand for PD-1, which is PD-L1. And this can be expressed on tumor cells as well as other cells within the tumor microenvironment. The interaction of PD-1 with PD-L1 on these tumor cells turns these T cells off. They can't actually kill the cell that they're targeted against. And so the idea of these next generation of checkpoint inhibitor therapies is that they are blocking this interaction of PD-1 with PD-L1. This removes that inhibitory brake on these T cells, allows them to become reactivated and ultimately kill the tumor cell. One of the key aspects of these therapies has been that as they have been coming to market, there have been approvals of biomarkers to predict if a patient would have a favorable response to these therapies. And really, there are three broad groups of diagnostics that are used here. One is IHC, which is shown at the top for PD-L1 itself. There are two molecular tests at the bottom that we'll discuss in just a second. So there are currently 3 IHC-based assays using different antibody clones against PD-L1. And these have really been around since about 2015 as these therapies came to market. I would mention that the work that was done with the 22C3 clone was done at discovery through the initial stages. And so really we were on the forefront of pushing these new checkpoint therapies to the market. On the bottom are the 2 molecular tests. The first one is looking for deficiencies in either those mismatch repair proteins or a degree of genomic instability by microsatellite instability. These can be performed either by IHC for those mismatch repair proteins or using a PCR-based assay for MSI. The final one is tumor mutation burden. This is an NGS-based readout that really came to market within the last 5 years or so. In the grand scheme of biomarker evaluation in cancer, these are still relatively new. And so there's still a lot of specimens in biobanks that would have either been processed or acquired prior to when these therapy or these biomarkers became standard of care, or potentially collected at sites where that wasn't the standard of care yet. And so really having a repository with these new markers, you have the opportunity to retrospectively go back and profile material that may be older, but still holds a lot of value in trying to push both the mechanisms of action and patient stratification of current therapies, as well as identifying new therapeutic targets. So we evaluated our DTC biobank to understand, could we use it and then further characterize it for these different biomarkers? So the first one we looked at was the expression of PD-L1 itself. Now, the clinical assay here is an IHC-based assay. DTCs no longer have that three-dimensional structure. They're no longer really amenable to IHC assays. So we needed to find a system that we could use to screen for PD-L1 that wasn't IHC-based. And so to do this, we performed a correlation study. On the bottom panels on the left here, this is IHC that was performed on tumors that we acquired where we also acquired fresh tissue that was dissociated and cryopreserved. And here I'm showing 3 examples. An example on the far left of a tumor that's scored by our pathologist as 100% PD-L1 expressing, one in the middle that scored as 0%, and then one in the right that showed at about a 28 to 30% range. For the DTCs that were generated off of these same tumors, we did multi-parametric flow cytometry and looked specifically at the tumor cells within those tumors, defined either by EpCAM percentage for all of our epithelial-based indications, or we use CD146 for melanoma, as melanoma doesn't express EpCAM. And then we looked at the PD-L1 expression on those tumor cells. We did utilize an isotype control, which you can see in the blue histograms here, just to account for any background staining that we would see with these samples. And what we observed was a very high correlation between the signal that we observed by IHC and the signal that we observed by flow cytometry. And we've done the study over dozens of samples at this point and shown very, very high correlation across all the samples that we've tested. At this point in the biobank, we've focused on 6 main indications that in the clinic are targeted by PD-L1 and PD-L1 therapies, primarily bladder, colorectal, head and neck, renal, lung, and melanoma. And we can see that if we look for the percentage of PD-L1 positive tumor cells in each of these, it really does vary quite a bit, with some patients having high expression, like these patients observed in head and neck and lung cancer, where others really in any cancer can have low or no expression of PD-L1. So we do now have a robust assay that we can use to screen our DTC biobank for PD-L1 expression for potential tumoroid development from those indications. The second biomarker we looked at was microsatellite instability for MSI high or deficiency in MMR proteins. Again, we've lost this 3-dimensional architecture in DTCs. And so while there are some methodologies to look at protein expression, we really focused on the PCR-based readout for this assay. And so in this case, we used the TrueMark assay provided by Thermo to look at microsatellite instability. So this uses 13 different loci, which are seen on the left here. And really we're looking for the acquisition of very small or very large fragments, which would indicate instability at each of these loci. We then profiled microsatellite instability across the 3 indications that it's most commonly observed in, which is endometrial, gastric, and colorectal. And this is anywhere from 25 to 30 samples within our gastric DTC bank to almost 150 in our colorectal bank. And we found, and this is consistent with the literature that's out there, that anywhere from roughly 20% to almost 50% of samples were MSI high when we observed the biobank. And so now we have even an additional level of characterization on top of this where we can develop models based on genomic instability. At this point, we haven't evaluated tumor mutation burden. That is actually an active area of research as we explore more NGS-based screening routes. And that will be part of the additional characterization that we're going to have in the future. So Phil had mentioned that we have a very robust biobank that we've developed, and this shows a snapshot of what that biobank looks at, and honestly what it looked like probably about 2 weeks ago. This is highly dynamic, and honestly, if we look today, these numbers would not be the same as samples are being consumed to go into different pipelines to answer different questions, and as new samples are coming in from our clinical sites to be processed. But what we do have is anywhere from a dozen samples for melanoma, which at this time tends to be one of the more difficult indications for us to source, to almost 400 unique patients in breast, endometrial, and renal cancer, as well as lung, to almost 750 unique patients in colorectal cancer. And these are now being profiled not only for those targets specifically to look at checkpoint inhibitor therapies like PD-L1 and MSI, but also for the presence of HPV, for example, in cervical and head and neck cancers, or the HER2 and hormone receptor status in our breast cancers. And so now we have a huge, very robust biobank that we can start pulling samples from to generate complex tumor models. When we focused on tumor models, we decided first to focus on colorectal tumoroids. There's a couple reasons for this. One is, as we saw on the last slide, it is by far the most robust of our inventory. So we do have a lot of samples that we can pull from this, as well as a lot of samples that can be profiled and have different genetic characterizations. Also, as we looked at the systems available for tumoroids, colorectal was one of the easier systems to develop. So it was a natural place for us to start. We started with about 10 to 12 colorectal DTCs. And what I'm showing here are 2 patient samples that we were able to take through to a full working bank. And I'll describe what a working bank is on our next slide. The advantage that we have, and that Phil described quite well, is that because we have EMR access and we get all the pathology reports, we know the location that this tissue was originally came from, what stage it was in, as well as all of the patient demographics, their age, their race, their gender. We also have tobacco and alcohol history, as well as other different metrics that we can pull on. Interestingly, for these two samples in particular, when we did look at them, both happened to be MS stable. So there was no evidence of increased genomic instability, as well as being wild type for a couple key oncogenes that we looked at. What you see below is actually two bright field images of the working banks of these tumoroids. They do have slightly different morphologies, but this allows us to assess both their size and the quality of the specimen as we're growing them. So we've got these two nice tumoroid models now, but really the first big question that we wanted to answer was, are the inherent genomic and transcriptomic profiles of the parent tissue maintained through tumoroid development? And so that's what we did. The first way that we did this was through bulk whole exome sequencing. And so to orient, as we build out this slide, what we're going to look at is we're going to look at the signature in the parent DTC. And this is going to be the entire dissociated tumor. So it's going to contain not only the tumor cells, but also the immune cells that were present, fibroblasts, endothelial cells, all the other cells that make up the tumor microenvironment. When our tumoroids are developed, we initially develop them into an embedded stage. So the embedded stage is a higher density of basement member extract. They all do form in little semi-solid domes. And it does provide a rich source of nutrients and close contact for our tumoroids to form. However, it is restricted in that these domes can take a while to form, and there's not really a way to scale them. So these embedded cultures are then worked up into what we call a suspension culture phase. In this case, there is still some basement membrane extract present. It tends to be at a lower concentration, but these tumoroids then grow more as a non-adherent cell line would. You can get a higher density per unit volume, and it allows for expansion. Because of these first two formats, we wanted to keep these very low passage because we are starting with a primary patient sample that's not undergoing transformation. And so these are very low passage banks that are frozen back and then only thawed and scaled up when we want to generate a working bank. And our working bank is really the bank that's going to go through all of the different assays that we are going to perform on our tumoroids. The other nice way to think about this is that should the working bank ever be depleted, we have a parent suspension bank that we can go back to make more working bank. Should that be depleted, we have the embedded bank to go back to. And so this allows us to basically have a very robust system generate more tumoroids from the same patient material. So this is looking at our whole exome sequencing. And so to orient, we're going to look on the y-axis at the variant allele frequency at each of these loci in the parent DTC. On the x-axis in each of these is the embedded suspension and working bank. And there was a high degree of correlation between the DTC, the parent tumor sample, and the embedded suspension and working banks. As these are developed, that correlation, if you look at the embedded bank versus the suspension bank, becomes almost nearly 1 degree, or 1, an R-squared value of 1, so very, very highly correlative, and that is maintained in the suspension versus the working bank. So what this tells us is that the genomic integrity is maintained from that parent sample all the way through to the working bank that we're going to use for our studies. We similarly did this on the transcriptomic side. So this is a poly-A-based bulk RNA-seq. We performed this again on the DTC as well as each of these banks that we've generated. And what we're going to look at here is the transcripts per million for each gene. Now, in this case, we do see that there's a slightly lower correlation. It's an R-squared value of about 0.77 to 0.78 when you look at the DTC compared to each of these banks. However, this is intrinsically not that surprising as DTCs include a lot of cells that aren't tumor. Again, those immune cells, fibroblasts, and endothelial cells. However, as we look at just the tumoroids themselves, from the embedded stage to the suspension stage, again, we're getting above 0.95 R-squared values, very tight correlation of the genes that are expressed in each of these banks. And it essentially is 1 when you get to the suspension banks versus the working banks. So now we know that both our genomics are highly correlative and highly stable, as well as our transcriptomics. So we feel confident that our tumoroids are actually replicating the parent tumor cells that we started out with. So the biggest question is we now have two lines, two colorectal lines. Every indication is that these lines are distinct, but we wanted to confirm. So we actually looked at our whole exome data and our transcriptome data from both the DTC and the working cell banks, but comparing the two lines together. And so when we looked at the comparison of these, we have the whole transcriptome analysis on the top. Our one tumoroid line is on the Y, our other tumoroid line is on the X. We did see a correlation at the transcriptome level between the DTC from each of these lines as well as the tumoroid. Probably, again, not entirely surprising. A lot of the genes that are being expressed are general genes for cell health, but there are a lot of genes that are differentially regulated, and we are looking at those genes right now and could provide some key information as to which of these tumoroids would be valuable for specific assays and specific research questions. When we did look at the whole exome data, however, there was no correlation between either the DTC sample or the tumoroid that was generated from them. The R-squared values are not correlative at all. And in fact, most of the unique mutations were present either only in one or the other of the tumoroids. There were some, but not a lot in both. So tumoroids are genetically distinct and not related to each other at all. So we've got a great data set. Now we wanted to ask, can we mine this to answer some initial very high level questions? And so we moved from our checkpoint inhibitor therapies to another therapy that's very active in the market right now, and that's antibody drug conjugates. So the principle about an antibody drug conjugate is that we now have an antibody molecule that is targeted against a putative tumor antigen or a cell surface receptor unique to tumor cells. But this antibody has been manufactured such that there's a linker that is attached to a payload. And this payload should be toxic to the cell that internalizes this antibody. So when this antibody is administered, it binds to its target on the cell surface. It is internalized, and in the lysosomal, endosomal compartments within the cell, those linkers are cleaved, the payload is liberated, and it can therefore mediate its effects on the cell. And the payloads generally will focus on any of three different components, either DNA itself, microtubules, or topoisomerases. And so there's a lot of active work in these antibody drug conjugates, and they're very specific to their targets. So can we start to bifurcate just our two tumoroid models by different expression of antibody drug conjugates? So this is a figure that's from a website that is very good. I highly recommend going and reading it, but it looks at the top 20 targets for ADCs that are under development. And this is from a couple of years ago, from 2022. And the reason I like this is it shows not only the number of ADCs that are going to the market or in various phases, but how many of them. For example, HER2, very popular target, a lot of work being done to focus on antibody drug conjugates against HER2. Now, these top 20 targets do have some non-solid tumor targets on them, things like CD22, CD19, BCMA. Those are focused more on our hematological malignancies, multiple myelomas, AMLs. But there are a lot of targets here that are specific to solid tumors, including a lot of active research, the B7 family members, B7H3 and B7H4, ROR1, Nectin-4, a lot of molecules where there's a lot of active research in them. So we've got this nice set of what ends up being about 15 to 16 ADC targets. Can we mine our data sets to see if they're expressed in our tumoroid models? So what we're looking at in this is our two tumoroid models, our 3309 model and our 1104 model. And we're looking at those ADC targets. What we're looking at here in the black is the log2 TPM for the RNA-seq data. We also ran unbiased mass spec analysis through our proteomics division to also get an idea of what the expression level is at the protein level. This proteomics data was very, very cool. We got about 8 to 9,000 unique proteins per tumoroid line. And it really gives us the ability to look at, is not only the mRNA present, but is the protein present as well. And so for the mass spec, what we're looking at is the log 10 intensity. What is very, very interesting right off the bat is that there's a high correlation between if the RNA is present and if the protein is present. In that if it's not present at all, let's look at Claudin 18 down here, it's not present by either metric. If it is present, if we look at EGFR, it's present in both. And there was really only one that we saw where the RNA was present, but we didn't identify the protein by mass spec. That's the folate receptor alpha. We're looking into this a little bit more to see if this is just a calling issue or if there is indeed no protein expressed in these two models. What was interesting was that there was one of these ADC targets that was differentially expressed, in that in the 3309 model, we saw evidence of both ROR1 mRNA and protein, but it is completely absent in the 1104 model. So even with just two models, we can also already develop a model system to study specifically ADCs against ROR1 when we know the target is present and when we know it isn't. So we have a robust data set. We've already been able to whittle it down to interesting targets. Can we place these in assays to start evaluating drug or potentially targeted therapy evaluation? So the first model we looked at was more of a bulk-based model. So we looked at cell Glow 3D. So in this case, we seeded our tumoroids 96-well plate and did an 8 doses dose response curve of 3 compounds. This is 5-fluorouracil, which is going to affect DNA, etoposide, which is going to complex with DNA and topoisomerase, and then finally paclitaxel, which is going to affect tubulin. These tumoroids were allowed to grow for 7 days in the presence of these various doses, at which point we measured ATP using the cell Glow model. And what was very interesting here is that we could already see just some basic differential responses. 5-fluorouracil, paclitaxel largely had the same kinetics in response to the various compound concentrations, while there was some slight differences in etoposide, in that at the higher concentrations, the 1104 model starts to die, while the 3309 model tends to remain pretty stable throughout all the concentrations that we tested. This is a great model system. It goes very quick. You can test a lot of different drug compounds and get data very, very robustly. But this is looking at a bulk model. And really, as we moved from compounds specifically targeting the tumor cells to those where we're looking at influencing the immune system against the tumor cells, we needed to move to a slightly different system. So what we have started to evaluate is the ability of these tumoroids to grow in live cell imaging systems. So this is an IncuCyte S3 instrument. These tumoroids were seeded back in domes. So you can see the presence of the dome here is the overall circle that these are present in. And these are imaged every 4 hours over several days. And what this can show us is this can actually show us that these tumoroids are in fact growing over those time courses. And because we can actually see the individual tumoroids, we can now add back other cells. We can redirect immune cells to these tumoroids. Do they kill them now? Or are they non-responsive? But now actually looking at the cells themselves almost at like a cellular level instead of as a bulk assay readout. So now we've got all of our models built, we've got assays built. Where are we heading in the future? And really the future is twofold. The first is we've got 2 colorectal lines that we've got very well characterized. That obviously needs to be expanded. And not only within colorectal cancer, where we're expanding those lines, specifically looking at MSI, but also for other indications. Lung cancer, breast cancer, ovarian, and head and neck are ones that are high on our list to test next. Interesting for ovarian, one of the models we're looking at is not necessarily starting with associated tissue or associated tumor, but starting with ascites fluid from ovarian cancer patients, as that material already would be somewhat acclimated to a more suspension type format. And as really the theme for this webinar has been, we're going to focus heavily on biomarkers when we're developing these new lines. We really want models that are PD-L1 positive and negative lung, HPV positive and negative head and neck, and then really breast models where we know the hormone receptor status, but that we've got specifically HER2 positive and triple negative breasts. So really we can be very specific on the models we're generating. And then the second is really pushing these cold culture systems. The next logical step there is to reintroduce immune cells. This could be potentially a hurdle where you've got essentially a purified tumoroid, but it has naturally lost its intrinsic immune cell component during development. The advantage of the cryopreserved DTC biobank is that there are still parental vials or parental material left. We do have some nice magnetic methods to pull out specifically T cells or full immune component cells from those samples, reseed them back into the tumoroid. In the absence of that, every time we do make a tumor collection, we try and largely succeeded also collecting whole blood from the same patient. So those also provide a nice component of the immune system test. And then finally is a more unique system. So in the process of doing the whole exome sequencing, we do have 6-digit HLA resolution across both the class 1 and class 2 loci. We also have a very robust normal donor clinic. The lab happens to sit right across the hall from the clinic. And this allows us to basically HLA match as close as possible the tumoroid's HLA status to our normal donors. And so in this case, we're using more of an allogeneic system, but with as much matching as possible. The other cell type that we're focusing on is cancer-associated fibroblasts. These luckily are a little more easy of a system to work with in vitro. If you take a parental DTC and essentially just let it lay down on a plate, fibroblasts will naturally just grow out. That's an example of what you can observe here on the right. And the nice thing about these fibroblasts is they're pretty robust. We've taken that fibroblast line on the right there, frozen it back, thawed it, replated it. It thawed out great, gave the same exact morphology. And so now we really can move from having a single tumoroid to having an immune system component added either from it or from a matched donor, as well as the fibroblast component as well. And we're trying to basically recapitulate the tumor microenvironment as much as possible using these long-term tumoroids as our starting material. So with that, I'd like to thank everybody for coming today. We can take some questions. Phil and I would be happy to answer them, and I'll pass it back over to Liam.
Fantastic. Thanks so much, Sean and Phil, for the great presentations. And with that, we will move right on to the Q&A. And as a reminder, if you do have a question that you'd like to ask, you could submit it using the Q&A tab on the right-hand side of your screen. All right, first one here. Have you evaluated the ability to genetically manipulate tumoroid models?
Yeah, I could take that one. That is a great question. That's really one of our future directions after we get more of these lines established. We are looking at this in two formats. One is one of the benefits of the IncuCyte system is its ability to read fluorescent proteins. So we are working to essentially have our tumoroids have GFP, have some of the nucleate red viruses that can be used, such they're a little more easy to evaluate under the IncuCyte system. And then second, that is moving into some of the CRISPR-Cas9 systems. Can we go ahead and add potential biomarkers or targets? Can we remove them a little more specifically and have some nice matched tumoroid models plus or minus different proteins?
Excellent. Yeah, great answer. All right, next one here. This person says they're interested in prostate and pancreatic tumoroids. Do you have any plans for those?
Sure, yeah, I can take that one. Yeah, it's great to hear feedback on what indications people are interested in. Sean just kind of went through some indications that we've either developed or are working on, including the colorectal. For prostate and pancreatic, those are a little more complex. They are definitely something that we're viewing in our pipeline, but don't currently have immediate plans to release those. I would direct people to the handout section to kind of take a closer look at our offerings, either on our webpage or through those materials, and to contact us directly with your request for various indications, and we can get back to you and work with you on timelines for those availabilities.
Excellent. Thanks a lot. All right. Have you considered either normal or non-oncology tumoroids?
Sure. This is kind of related to the last question, right? What other indications can we do? And as I mentioned kind of at the beginning of this webinar, organoids started out with normal tissue. It was kind of the first steps on getting to tumoroids. Here at Discovery, we specialize in diseased specimen collections. We also specialize in normal collections, but for our Tissue components, a lot of that comes from oncology focuses. So that's kind of where we're starting and putting a lot of our R&D work in expanding out the entire suite of our oncology offering. From there, we're going to look more at the normal capabilities, but once again, that's further down the pipeline roadmap, not something that will be immediate.
Excellent. All right, this person says, great talks. Do you have functional killing assays with immune cells?
Yeah, I can take that one. Currently, we're developing the data there. As Phil and I meet basically weekly to talk about tumoroids, we have quite a lengthy list. That one's pretty high on the list to do. We're probably going to start we have some isolated NK cells from our apheresis collections. We also do some very large-scale apheresis collections. That tends to be a low-hanging fruit to start to develop those before we move into the actual precious patient material, if I could say that. So yes, we don't have it done yet, but it is pretty much number 1, 2, and 3 on the list of to-dos.
Excellent. Thanks a lot. It looks like that's actually all the questions that we have. And so maybe we'll call it there. But if you did have a question that you didn't get to ask, definitely be sure to contact info@dls.com if there's something that you want to know, and the experts at DLS would be happy to help you out. But yeah, thanks so much, Sean and Phil, for your really great insights today. It's been a pleasure having you with us.
Awesome.
Thank you, Liam. Appreciate it.
Awesome. And in closing, we hope you enjoyed this Discovery Life Sciences and Scientist.com webinar, and we'll see you again next time. Have a great day, everyone.